Good evening, everybody. Welcome. My name is Corky. I'm with Yolo Basin Foundation, and this evening for Flyway Nights, we're going to be hearing from our fellowship recipients about the work that they have been doing in the bypass and the research going on. So tonight, we're going to be hearing from Stephen about his work with the distribution of the giant garter snake and the western pond turtle. From Brian Lee, we'll be hearing about his work with radar and how he can use that to find out more information about the bats, what they're doing, where they're going. And from Mackenzie, we're going to be learning more about the salmon of Pewter Creek. I also want to remind you that next month, we will be learning about the Big Notch project at the Fremont Weir. And if you're not familiar with that, the Fremont Weir is located to the north of us, where the water from the Sacramento River spills over the levee, a low place in the levee, and brings that water into the bypass. And the notching of the Fremont Weir will bring water in sooner and hopefully will benefit the fish. So now on to our fellowship recipients. And we are going to start with Stephen. And let me spotlight Stephen here and have you take it away so we can learn more about what the work you've been doing.
All right, let me. All right, you see it?
Yes, it's up. Thank you.
Perfect. Okay. Good evening, all. Thank you for the introduction, Corky. As you said, my name is Stephen Gergeni, and I'm a graduate student at California State University in Sacramento. I am pursuing my MS in biology. My advisor is Dr. Ron Coleman. And I would like to start off by saying thank you to the Yolo Basin Foundation for their generous support of my research. And also thank you for the opportunity to share my research with you tonight. So my study is population distribution and habitat assessment for giant garter snakes and western pond turtle within the Yolo Bypass Wildlife Area. So understanding species distribution is necessary for effective conservation and ecological management. There is importance to continually study species at the population level so you know what they're up to at any one time. This study is designed to update data for species presence and distribution for the giant garter snake within the Yolo Basin Recovery Unit. The giant garter snake presence and distribution data from the study area I'm using is dated, having last been assessed more than a decade ago through a series of physical trapping methods. So what are they doing now as opposed to then? Also, distribution data will be collected for the West Coast endemic western pond turtle as part of the sampling effort. The turtle is a species that is of conservation concern to wildlife managers, and local distribution data appears to be somewhat lacking. Additionally, the study will attempt to determine if the distribution patterns of the snake specifically differ in comparison to historical wet-dry cycles of the recent past to the current dry-dry cycles of today. So California's Central Valley has undergone significant anthropogenic reshaping over the past century and a half. Habitat loss and fragmentation are the leading cause for the decline of most species on the United States endangered species list. So as climate shifts, wildlife habitat will continue to be altered by or lost to these changes. This makes identifying and protecting unique landscapes, habitat, and species community vital for the future of the state's wildlife and plant biota, a significant portion of which is endemic to the region. Continued monitoring of habitat conditions for the recovery of listed species is an often integral part of each species recovering part. So yeah, this image, it's existed for a while, but it's still always, every time I see it, it's pretty shocking on the changes. One such unique location is the Yolo Bypass, and it's located in Sacramento and Yolo counties. The Yolo Bypass is an extensive flood control system for Sacramento and surrounding communities. This complex channels water from the Sacramento River around and away from the heart of Sacramento during high water events. The bypass exists within the region's historical river floodplain and retains a historically similar hydrological cycle to the pre-settlement and river delta systems, as well as continuing to provide 59,000 acres of a contiguous habitat for some of California's most sensitive wetland species. Located within that is the Yolo Bypass Wildlife Area, which we all know and love, which is an approximately 17,000 acre area within the middle to southern end of the bypass. And it is frequently inundated with up to 15 feet of water for multiple months during California's wet Mediterranean winters. And this is an old map, but this is, what it looks like. And this is probably like the first map of the wildlife area, but now it's reaches all the way down to here. So by virtue of design, the Yolo Bypass provides valuable habitat for many of California's sensitive and dwindling aquatic species, such as the giant garter snake and western pond turtle, making it a perfect location for habitat use assessment surveys. So I would like to give a brief background on the target species before moving on. So the giant garter snake, it's a really pretty little guy here. And so giant garter snake, or GGS for short, is a non-venomous aquatic snake that preys on a variety of native and non-native fish and amphibians. They are the most aquatic species of snake that are native to California, and they are also endemic to the state, meaning that they are only found in California. They take prey in or from the water and are active during the day and at night whenever it's during the warm season. Their active season is approximately from April to October. They're ovoviviparous, meaning they carry their eggs internally until they hatch and then they give birth. Mating occurs in mid-spring and then birth sometimes in the late summer. They can reach a total length of 162 centimeters or just over 5 feet. And unfortunately, they are listed as threatened on both the federal and state endangered species list, mainly due to lack of habitat. Historically, the giant garter snakes were found along the edges of large flood basins, freshwater marshes, and tributaries from Butte County to the north and Kern County to the south. Today, their range extends from Butte and Glenn counties to the north to Fresno County in the south, where they are known to live in a variety of agricultural, managed, and natural wetlands. These snakes also live in artificial waterways and agricultural wetlands like irrigation and drainage canals and rice fields and the adjacent uplands. At this point, only about 5% of its historical wetland habitat acreage remains. So, yeah. The habitat components, which have been identified as the most important to giant garter snakes during their active season, include a freshwater component with protective emergent vegetation, which provides cover during forage and an upland component near the aquatic, which can be used for thermal regulation and shelter. Freshwater marshes, flooded rice fields, slow-moving sloughs are all examples of suitable habitat. These are all pictures of various places in the wildlife area. During the winter brumation period, their habitat includes uplands with animal burrows, riprap along levees, and waterways, waterway banks well above ordinary high water levels. The snakes will sometimes travel hundreds of meters from water to find winter refuge. So Western pond turtles are a small to medium turtle ranging between 3.5 to 8.5 inches in shell length as adults. Hatchlings are approximately 1 inch in shell length. The species is dark brown to dull olive in coloration with yellowish undersides. The top of the shell is typically wide and the edges lack serrations. Males have a yellow throat and they do not have the red ear of the aptly named red-eared slider, which are an invasive species from the eastern US. So this is a handsome fellow here. So the Western pond turtle was split into 2 species semi-officially around 2014. The northwestern pond turtle and the southern pond turtle. They're California's only native pond turtles. The northwestern pond turtle ranges from roughly San Francisco Bay northward and then along the Sierra foothills. Again, this species is listed as a species of conservation concern with Fish and Wildlife, which provides the turtle with some legal considerations. For the majority of the year, these turtles are associated with permanent or intermittent waters with good basking sites. They can also be found in upland habitats adjacent to water bodies during the nesting season. Migration and overwintering seasons, during the migration and overwintering seasons, the turtles sometimes leave water to find a better place to live or mate or lay their eggs. So my study locations when looking for these species were based on a few prior surveys. So from 2001 to 2012, fellow giant garter snake researcher, Mr. Eric Hansen, performed extensive studies for giant garter snakes in and around the greater Yolo Bypass and Atomas Basin areas. His studies are marked with all of the study areas are marked with all of the black dots. And the turquoise coloring denotes all of the giant garter snakes observations in the selected area up to and including 2009, as reported to the California Natural Diversity Database. So you can see a lot of studies were done here on the wildlife area in and around 2009, and they were finding them. So I'm trying to see if they are still here or if they are there and other places on the study area. So whenever it came to the Western pond turtle, though, when querying species records from the Natural Diversity Database, it appears that official observation data appears to be lacking. The orange polygon shows a partial overlap of my study area with the turtle observations up to last week being in pink. For anyone that has spent any time in and around the Sacramento area's rivers and wetlands, it is obvious that the turtle observations are underrepresented. So the California Natural Diversity Database isn't the end-all be-all of distribution data, but it does collect a lot. And so this is kind of a snapshot that I was taking. So as you can see, maybe we should take a little bit better look in this area to see who's there and how many of them, or at least how spread out. So whenever I was choosing sample locations for my study, the most important consideration was the presence of historically permanent or semi-permanent water. As a part-time scientific aide for CDFW at the wildlife area over 7 years, I am familiar with the general distribution of water throughout the year during both flood and drought. So I was able to discount areas which may appear to be prime, but which that I'm aware will be dry by midsummer. The wildlife area, for the most part, naturally changed from north to south and west east, with most permanent water during the spring and summer being due to active management for agriculture or grazing, or in sloughs or ponds designed to capture the winter water or stormwater drainage. I chose 13 locations, sampling sites, which exhibited good habitat components for both species, with a slight weight of consideration be given to giant garter snake habitat. So 12 of the areas are within the bypass levee system, with the remaining being just outside the levee, but still within the wildlife property. So like the green there is that little square that sits up there outside of the levees. But so the actual interesting part here, it is scientifically important that research methods continue to evolve such that studies are using the best practices available for their tasks. Historically, species presence, absence, and distribution research often necessitated the use of visual encounter surveys or extensive trapping methods. These techniques are frequently time-consuming, expensive, and require a large field crew. Additionally, restrictive or hazardous terrain may factor into feasibility. Furthermore, trapping and handling of animals involves specialized training, ethical considerations, legal requirements like permits, as well as posing a danger of harm to the species being studied. Lastly, the studies of organisms which are cryptic or difficult to detect because of small numbers, small size, or acute avoidance behavior pose additional challenges. The giant garter snake is a cryptic species prone to silently slide into the water to hide in aquatic vegetation before a researcher is near enough to notice. Additionally, in most of the giant garter snake's range, it coexists with two other garter snake species with similar markings that are also known to flee detection into adjacent bodies of water, making definitive visual identification difficult. It is also difficult to identify some field markings when the snake is half submerged, obscured by vegetation, and or covered in mud. This picture here has two snakes in it, by the way. Accurate identification usually means handling, which in the case of the giant garter snake requires a federal recovery permit as well as a California scientific collecting permit. And the turtles are not much easier to capture. My method for presence absence sampling is to use environmental DNA or eDNA. Conveniently, sampling techniques for the study is applicable to both species, so having two collection methods was not necessary. The benefit of this method is that it is non-invasive, meaning that a collecting permit is not required. It can be used to determine the presence of low habitat density species, cryptic species, or species in difficult or dangerous terrain. It can save multiple studies hours if all that is needed is presence and absence data, and it makes for an excellent means to focus any future trapping efforts to areas with a greater chance of success. eDNA is a relatively new data collecting technique that tests for the presence of a target's species DNA, which has been left in the environment by the creature's passing. This could be sloughed off skin, feces, urine, mucus, etc. Testable media can be water or soil or even air. For aquatic species such as the giant garter snake and western pond turtle, water from likely habitat locations is collected, then drawn through an inline filter which traps any detritus suspended in the water that potentially contains the DNA left by a recently passing snake or turtle. Here you can see me collecting water at one of my sampling locations. In the case of a linear water body, such as a canal or slough, I attempted to get samples at areas where the water was swirling so that a suspended particles from upstream would be mixed all together for easy collection. About a quart of water at each location was collected once in mid-April, then mid-June, then August. These times represented the dates when the snakes would be first emerging from their winter burrows and looking for mates, then would be actively hunting and dispersing, and then during their birthing season. Using a cordless drill to power a peristaltic pump, I drew the collected water through the inline filter which is over here, until the filter clogged. I then placed the processed filter in a separate baggie inside a cooler. For each collection location, I processed the water sample in triplicate. This resulted in approximately 120 samples over the course of a survey year. The filters were then tested in the lab against a known control for the DNA presence of both species. Initially, I only tested a single sample per location per time period in order to keep my budget streamlined. This is the first year of a 2-year study, and after both my study years are complete, I can go back and test as many samples as possible in duplicate or triplicate as funding allows. That way, I have a broader set of results. So at this point, after all of this, I have detected western pond turtles at 3 locations within the study area, marked as blue and green, and at giant garter snakes in 2 areas, so in green and yellow. And so one detection was in the historical giant garter snake, known location, at least from the prior data. And then the other one was from the southernmost end of the wildlife area property, where was a new to the study area detection. So that was an interesting. So this is a couple of examples of the data reports that I get back. So this one on this side is saying that out of all of the samples I took, I didn't get a detection. These are my controls, my positive controls, to make sure that the lab's data was working. I found one turtle example here. And then on this side is whenever I finally found some garter snake activity. So here was on this location, and this is that northern one, and this is that southern location. This one here was, I was lucky enough to go out and survey another area that I knew in another location, not associated with my study, that had giant garter snakes in it, fairly thick. And so I just was making sure, because this one is in August, I was making sure that my collection technique could get a positive result. So I was happy to see that I got a positive result from a place that snakes are known to be fairly thick. So I was doing it right. So what I've learned so far, there are giant garter snakes and western pond turtles present in at least some of my sample locations. My sampling techniques do provide for positive detection. There are giant garter snakes in historically occupied areas still, and there may be giant garter snakes throughout the entire wildlife area. So that was exciting. And then what's next? Well, I have a second field season in the spring and summer of 2023, where I will go check the same areas and see what I can find there. I will be spending some time searching for additional survey data in and around the Yolo Basin area to see if I can find any other studies. I mean, there's some out there, to see where those locations, what those locations are finding. Hopefully acquire some additional funding so I can run some more samples in duplicate and write a thesis and provide management suggestions based on where these snakes are currently and maybe in the future. And my acknowledgements and thanks, obviously the Yolo Basin Foundation for their very generous funding, and then all of the rest of the people that have been instrumental in helping me move forward so far. That's it. Any questions?
Yeah, we do. We have some questions that have come in. Brian wants to know, how much time do you have before the DNA sample is no longer able to yield information via eDNA?
So it depends on a lot of factors, but my understanding is it's good between 3 days and 2 weeks, depending on the clarity of the water, what amount is getting dumped, how much is basically being but how much is being dumped. I know that it doesn't last as long on the ground because DNA and UV light don't get along. But the idea is it's somewhere between 3 days and 2 weeks.
Great, thank you. And can the eDNA tell anything about the health of the animal?
Not to my knowledge. I know that they're getting better at it. Right now, you can't really, it's really difficult to tell the difference even between individuals at this point, from my understanding, because all we're looking for is the parts of the genome that are associated with all giant garter snakes versus the ones that look similar, like the valley garter snake. So what the hope is for like the eDNA is that to answer that question would be, you would figure out where you're getting the snakes, where you're finding the snakes on this, 16,000 acres. And then if you wanted to test for health, et cetera, then you would know that I should trap here instead of all of these other places because I'll have a better chance of finding them. But as far as I know, you can't really do health at this point because you're not looking at the entire genome or the condition of the genome. You're just looking at, is this piece specifically just giant garter snake? Does that make sense in a long rambling type of way?
I got one more for you, and then we're going to move to Brian. And then where did it go? Oh, can eDNA indicate anything about the density of a population?
I would think that you could make inferences by how often you detect. So I'm trying to make sure that whenever I do it, my areas are kind of, the water is all coming from one cell, at a time. So I'm not potentially picking up the same snake twice by he was in this, he or she was in this thing of water and then moved across to this other body of water, and they both drain into, so you could set it up to do it that way, but you need a lot of samples. So yes, but it would be expensive.
Great. Thank you. Thank you, Stephen. If you can bring your screen down, then thank you. We are going to move on to Brian here. Thanks, Brian.
Great. Well, thank you again, folks, for having me, and thank you again for the very generous funding. My name is Brian Lee, and I'm a spatial ecologist and a PhD student here at UC Santa Barbara, advised by Dr. Ashley Larson. And my research utilizes remote sensing and causal inference techniques to explore the interactions between food systems and natural systems. Although my background in research is originally in forest ecology and spaceborne remote sensing, at UC Santa Barbara, I've been able to focus my attention on agriculture in California. Now, California is the fruit bowl of the world. Every year, California is responsible for millions of tons of food, equivalent to $60 billion. And this is only possible through the massive use of pesticides. Pesticides that cost millions of dollars in manufacturing and application, are known carcinogens harmful to human health, and unfortunately decimate pollinator populations. But hopefully we don't have to rely completely on pesticides. Recently, I've been exploring a really exciting project looking at bats. And as a side note, and something of a confession, I am not a bat biologist. And I was initially hoping to use the occurrence of bats at Yolo to create a model detecting flying species using spaceborne radar, specifically flying locust using spaceborne radar for a food security project. But over the course of this project, I realized that bats are super cool. And so like the first chapter of my PhD has kind of shifted to this project. For example, a fun bat fact, the average Mexican free-tailed bat, which weighs approximately 10 grams, can fly 100 miles while eating almost 2/3 of its weight in pests. If we scale that up to the 500,000 bats in California, we're talking about 4 metric tons of insects eaten every evening. That's enough to fit the inside of a U-Haul truck. Now, bats have the ability to make a profound impact on pests and pesticide use. Unfortunately, the species is threatened by habitat loss and white nose syndrome, which has wiped out millions of bats across the US. This leads me to my project, Hungry Bats and Happy Farmers, quantifying impacts of bat foraging on California's agriculture. Now, we've already established that bats are small, fast, and difficult to track, especially because they fly at night. We know they eat a lot of insects, but knowing exactly when and where this occurs has been a traditionally difficult task. And hopefully my work will help fill some of these gaps and help solve some of the mystery. And this is where weather radar, machine learning, and cloud computing enter the scene. Similar to how the National Weather Service uses weather radar to track clouds, we can actually do the same to track bats. Now, radar data are notoriously difficult to analyze and process, and we use a number of different radar measurements to identify bats in our data. For example, reflectivity, velocity, differential reflectivity, spectrum width. These are just a number of radar data products that we need to kind of parse through to identify bats. So I first download these data, which are grainy and processed and noisy, as we see in this image. I then combine these data for ingestion into my machine learning algorithm, which means I kind of just take all these data, standardize it to a uniform format, and dump it into what I call a data cube, or a multi-dimensional array of data. And this makes the data almost indecipherable to the human eye, as we can see here in this data composite. I then take these data and feed it into my machine learning model. And this algorithm helps me identify pixels that are bats versus those that are not bats. I specifically use a deep learning algorithm called an artificial neural network. And I've been able to experiment with a number of different machine learning approaches, including random forest and SVMs, but ANNs seem to yield the best results. Finally, I aggregate the data of classified bat presence and absence to different timescales, where I'm able to see where bats are aggregating over a single evening versus where they're spending or aggregating their occurrence over years and hopefully even decades to explore and study patterns of bat movement and bat foraging across the landscape. The major challenge is scaling this data pipeline up to classify and aggregate terabytes of data over multiple foraging seasons. So here's an example of bats leaving their roost at sunset and fanning out across the landscape to go on their foraging mission. This GIF captures the first 2 hours of bat emergence in August 2017. And the radar tower collects data every 3 minutes, meaning there are 40 frames within this GIF. I'd like to highlight a couple colonies, including the Yolo colony outlined here. And here's just a picture from the Yolo Basin Foundation that shows you what this emergence looks like on the ground. Additionally, we see emergence from large colonies in the northeast of downtown Sacramento. Here they're flying out of a railroad bridge over Dry Creek. And when this GIF reloads, here I highlight the Casumnes colony that flies out of and roosts in Casumnes Preserve. But as we could see, as this GIF reloads over and over, there are just dozens of roosts in places where we see bat emergence across this kind of small landscape, which is really exciting. And so just to highlight my current progress and where the project stands, I've been crunching a lot of data. Each year contains approximately 4 terabytes of data, which is then condensed into a single 15 megabyte data layer that helps us identify areas of high bat foraging. Now, obviously, this is a lot of computation that needs to be done, and I rely on cloud computing in the form of Microsoft Azure. And Microsoft's AI for Earth in Azure has been an incredible sponsor of this research in the form of their technical expertise. As you can see, I've been processing data over a number of years. I've processed 2017, 2018, 2019 data, and eventually would love to come up to the present day. And this is because I'd love to explore how bats react to different shocks and events across these years, such as drought and wildfire. Eventually, if we nail down more funding, it would be really cool to see how bat dynamics evolve over time, starting in 1990, which is when the tower was first installed here outside of Davis. And I'm currently in the process of writing up the methods of this project for submission to a journal, and I'm shooting for methods in ecology and evolution. And as a side note, I'm planning on making all these data, pre-trained model, and scripts openly available for anyone to play with and analyze. And finally, I'm beginning to analyze the interactions between bats and different environmental variables and factors. Now, this image helps me identify areas of high bat activity over the course of a week in August 2017. We first noticed that there are many peaks that include bat roosts, such as the Yolo colony, the railroad colony, and Casumnes colony. We start to notice smaller peaks that include areas where I believe foraging is occurring in the foothills here, around the delta, and in the rice fields to the north. And I'm now starting to process pesticide use data to see kind of the effect of pesticide use and the bat response, or vice versa. Looking at how crop type and land cover type affects or draws in bats, and also weather and a number of other variables. I do this because I'd like to fully parse out the causal implications of these variables. And I'm planning on borrowing techniques from causal inference and econometrics to fully explore the relationship between pesticide use and bat occurrence. I think having this precise, quantitative, and detailed data makes all the difference when it comes to changing minds, or maybe even changing policy to support bat conservation. It's one thing to say that bats are eating a lot of insects, but with the technology to show people when and how many insects they're eating, perhaps, this might provide a lot of incentive for us to conserve bats. Ultimately, I believe it is essential that we communicate the importance of bats as pest managers and ecosystem service providers. And I'm hopeful that my algorithm can help us accomplish that. With that, I'd just like to thank folks here at Yolo Basin and folks who supported my research over this last year, including Microsoft, the Schmidt Family Foundation, Yolo, and Layla Harris, have provided a lot of training data that I used for training my model. And also Gary McCracken out of Tennessee, who's provided a lot of expertise on bats themselves. So thank you.
Thank you, Brian. So we have a couple questions that have come in. One is, how far do the Yolo Causeway bats fly from their roost when foraging? I've seen them emerge dozens of times and they seem to fly far away.
Yeah, absolutely. Thanks for the question. Let me go back to this GIF. I mean, even from this like 2-hour time period, we see them flying 10, 15 kilometers. And I haven't tried tracking a single pixel across my entire study area. I mean, that would be a fun exercise, but I just haven't done that yet. My understanding is that they fly quite far during their foraging routine. And the kind of census is that they fly up to 100 miles, 50 miles round trip. And so I wouldn't be surprised if we see that here as well. And I think there are others that might be better able to answer that question. But within my data sets, I've noticed them flying quite far. I just don't know how far.
Okay, next one is, have you correlated bat use and specific ag crops?
Yeah, we're actually in the middle of doing that right now. I think some of the grain crops were coming up is really high in addition to rice. But we've literally just met to process some of the land cover and crop data like this week. And I think the early results were showing grain crops and rice. But again, we'd have to get into the weeds with the data before confidently saying that. And we're hoping to write that up soon too.
Excellent. Well, thank you, Brian. Thanks for all the information. Pretty exciting. And I know there's some bat peeps out here that are watching and interested in what your work is doing.
Great, thank you.
Yeah, absolutely.
Okay. And you guys can hear me and you can see my, what was that?
Yes, it looks like you're all set.
Okay, great. Thank you so much. Thank you, Corky, and thank you for hosting tonight. I'm really glad to be able to share with you all this evening what I hope are some interesting aspects of the research I did while working on my master's at UC Davis. At its core, the project that I work on is an ecological monitoring effort born of mitigation requirements. But my final objective was to characterize the process of Chinook salmon reconciliation in Lower Poudre Creek and identify any limiting factors to recovery, if any were present. And so, as Corky mentioned earlier, I have presented here before, so I hope I'm not repeating myself. But today I plan to present to you some specific data that to me signifies ways that we can continue to build on the legacy of success that is Poudre Creek salmon. This evening, I'll mention a term resilience or resiliency a few times. And what I mean by that is the ability of an ecosystem, population, or organism, or salmon run to withstand and recover from disturbance. And so I'm sure, as many of you already know, the historical ecology of Poudre Creek is unlike that which we observe today. Poudre Creek today is constrained by 3 dams. The Monticello Dam, which forms Lake Berryessa, the Poudre Diversion Dam, which forms Lake Solano, and a small agricultural impoundment dam, or a check dam as I'll refer to it, that's located in the Yolo Bypass. There is no permanent fish passage at any of these dams. However, these wooden flashboards at the check dam are removed seasonally in the fall to allow for upstream passage of adult Chinook salmon. And just to orient you a little bit here, this, I hope you can see my cursor, is the Monticello Dam. Here is the Poudre Creek Diversion Dam, and the check dam is located about here. And so because there's no upstream passage beyond the Poudre Creek Diversion Dam in any time of year, all of the work that I've done and the data that I'll be presenting to you has been collected in this segment of the creek down here. So anything downstream of the Puda Diversion Dam. Thankfully, today the creek's flow is closely monitored and barriers are managed to benefit Chinook salmon. And since the early 2000s, these restorative actions have seen a lot of success. Salmon do generally return to Puda Creek every year to spawn, and we're able to observe their progeny or their babies in the creek every spring. However, I do think that it's easy to kind of fall into the trap that would lead us to believe that we're done and we did it. Salmon are present in Puda Creek all the time. But while they're going strong now, I think there are things that we could do to strengthen the salmon run in Puda Creek and help build ecosystem resiliency, such that in the face of climate change, drought, what have you, salmon continue to persist in Puda Creek. So in the next few slides, I'll share with you a few examples from our data that I think highlight areas of opportunity for us to promote a more resilient salmon run and continue to build on this legacy of success when it comes to Puda Creek salmon. So in monitoring this run of salmon, we generally perform adult carcass surveys to monitor the adults, and we trap juveniles, and we monitor the salmon as they return, and then the juveniles as they leave. We also monitor freshwater residents of the juvenile life stage, but that's kind of beyond the scope of tonight's presentation. But when we perform adult carcass surveys, in part, it gives us a rough idea of how many adults are in the system and where they are. But despite consistent effort across years, the number of salmon returning to the creek varies notably. And there can be a lot of reasons for this, some of which have nothing to do with Puda Creek, but obviously I'm going to focus on Puda Creek specific data. Most of the time in Puda Creek, these numbers and their variability are a reflection of when and how the fish can access Puda Creek, oftentimes as the result of passage barriers, whether they be space or time dependent, that prevent salmon from moving upstream. Here, for example, is a visual representation of the spatial distribution of recovered carcasses in Puda Creek in every year since 2016. Really quickly, the white and orange locations on the map, which are kind of hard to see, are hot spots or areas of greater density of recovered carcasses, and blue is areas of fewer carcasses. And as you can see in the early years of this study in 2016, 17, and 18, most carcasses were recovered upstream and then kind of petered off as you move downstream. These habitats have seen the most restoration activity and are generally characterized by improved spawning habitat. But in recent years, like 2019, 2020, and 2021, for a myriad of reasons, salmon migration upstream seems to have been interrupted. To me, these later years kind of serve as a case study in many of the ways that we can better manage the salmon in Puda Creek. So what I'm going to do is highlight these latter years of data that to me represent a few of the limiting factors present in the data. And I'll just note really briefly for 2019, pictured at the top here, that only one carcass was recovered late in the season. And I suspect that it's likely the result of a much delayed removal of those wooden flashboards that I showed you earlier. Unfortunately, there were some troubles in 2019 with pumps that help move water around in the bypass. And as a result, the boards remained in place longer than they normally would have. And so I think that Puda Creek just missed a lot of the returning adults in 2019. So that would be an example of a kind of a temporal limiting factor. But in the fall of 2020, after the initial recovery of carcasses from upstream reaches in November, all of a sudden we stopped seeing salmon arrive upstream. And as it turns out, the formation of a natural passage barrier, the really large beaver dam that I have pictured here, prevented upstream movement. So when flow was higher earlier in the fall, it was adequate to allow salmon to pass the dam, but as flow receded, it became impassable. In this sense, this beaver dam became a problem for salmon, both spatially and temporally in the fall of 2020. When measured, this dam was about 2.5 meters tall. And so it's likely that this dam existed in previous years, but grew to such a critically large size in 2020 that it finally became a problem. And for fall 2021, as many of you likely heard or experienced, recovered carcasses were found in the lowest reaches of Poudre Creek after poor water quality conditions resulted in the pre-spawn mass mortality of a lot of Chinook salmon. On the right is a chart displaying dissolved oxygen concentrations near Lisbon Weir, which is just downstream of Poudre Creek's confluence with the tow drain. And this is in each year that carcasses were recovered in Lower Poudre Creek, and the vertical black lines indicate the date of peak carcass recovery, or the average date when most carcasses were recovered. And you can see here the blue line that represents dissolved oxygen, and it has this precipitous drop in late October, early November in 2021 that likely caused the large fish kill. And the horizontal dashed line is a dissolved oxygen threshold pictured here at about 7 milligrams per liter. And generally speaking, 6 or 7 milligrams per liter is kind of considered a good threshold for dissolved oxygen for adult Chinook salmon. And you can see here that in the tow drain anyway, which fish have to pass through to get to Poudre Creek, we already kind of walk that 6 or 7 milligram threshold in every year. So that when there is a disturbance, in this case, possibly an increase in organic matter in the system, there's not a lot of room for environmental buffering. And then so on the left is just a picture of one of the adult carcasses that we recovered in early November that year. And you can see that it's a female that wasn't able to spawn and likely died during that anoxic period. So to me, this water quality barrier is another example of a spatiotemporal effect. It greatly restricted the space in which salmon could be for a period of time, and it was the result of just a few variables interacting in a big way to cause these low oxygen conditions. But whether caused by natural or artificial barriers like beaver dams or water quality or man-made dams, these spatial and temporal restrictions of spawning migrations for adults could have implications for the recovery of salmon in Poudre Creek. For example, migratory delay can negatively influence the condition and ultimately the spawning success of adults by depleting energy reserves and delaying arrival at spawning grounds. And time-dependent barriers, so those barriers that require action by us or require increased precipitation for them to pass, prevent passage, and also have the potential to bias the genotypes contributing to juvenile production, and could ultimately truncate the evolutionary timing of spawning migrations. All to say that by providing salmon a wider migratory window in Poudre Creek, we may be able to hedge our bets, if you will, to help ensure a greater diversity of run-timing genotypes contributing to juvenile production, and kind of bolster the population against these one-off calamitous events like the anoxic conditions in fall 2021. Okay, so while I think adult salmon often receive the most limelight because they're awesome and charismatic, understanding the dynamics and possibly the limiting factors present in the juvenile population is also important, because while rates of returning adults are critical for establishing a salmon run, survival by the juveniles is how we maintain a salmon run. In Lower Poudre Creek, we assess the fry and smolt stages by capturing them as they move downstream in a rotary screw trap like the one pictured here. And while I won't get into the juvenile trapping data in this presentation, I really want to highlight how we monitor them as they leave the system. So after we capture them, this is when we can perform an acoustic telemetry study. So to perform an acoustic telemetry study, we deploy autonomous receivers like this one pictured here, and we tag the fish with a small transmitter that has a unique identifier, and this is an example of one such transmitter. And then if everything works, that little transmitter will communicate with the receivers that we've put in specific locations, and that receiver will store the date and time that that fish with that transmitter passed it. And so in the spring of 2021, that's what we did. I wanted to characterize residence patterns and describe the possible passage barriers that we had identified that previous fall and the role they play in juvenile outmigration. And so you can see here where the receivers were placed. They're the black triangles in the fall or in the spring of 2021. The creek was segmented into about 7 reaches using receivers at the top and bottom of each reach. And possible passage barriers were identified as being that really large beaver dam that I pictured earlier, a road culvert, and that check dam that's in the bypass, the Yolo Bypass. And at each of those possible obstructions, only the check dam really stood out as impeding downstream movement by juvenile salmon. Passage rates at the large beaver dam and at the road culvert were about 77 and 75 or 76% respectively, whereas only 20% of the individuals arriving at the check dam were able to pass it successfully. And although understanding overall outmigration success is critical in Poudre Creek, it's also important to understand how fish are utilizing habitats within the creek so that we can begin to focus our efforts in areas that may represent mortality sinks, for example. So this plot, also looking at telemetry data, looks a little bit closer at the in-reach behavior of fish. Each reach is identified on the x-axis, and the time spent in that reach is on the y-axis. From this plot, we can see that the amount of time spent in each reach varied widely, but that observed residence times were longest in that most upstream reach. And increased residence time in this reach, or in general, I think could indicate holding patterns by juvenile salmon prior to outmigration. But after that first reach, you can see fish moved a little bit more consistently downstream, spending the least amount of time in segments D and E. And just to refresh our memories on the map, overall, it took salmon about 7 or 8 days to go from the point of tagging, which was up here at the rotary screw trap, just downstream of Winters, to travel all the way down almost to the tow drain. That last receiver was positioned downstream of the check dam. But most of the data here can be explained by the length of the reach. Generally, fish spent more time in longer reaches. That makes sense. However, if we standardize this data by looking at ground speed, we can see that transit rates were consistent, except for in segment D, right here. In segment D, average ground speeds tripled. But we know that the flow in segment D isn't particularly fast, so it's probably not the flow pushing them along. But from other studies we've conducted, we do know that segment D is broadly characterized by a low gradient, warmer water temperatures, and an increased abundance of non-native predators, like largemouth bass, for example. Therefore, I think that this increased ground speed in segment D could be caused by a lack of holding habitat or by the presence of predators. Now, unlike the other examples, like the dam that I showed you, this data doesn't necessarily tell us that something is wrong in segment D, but it indicates to me that there's something going on here that we should look at. All this to say that from the data that I have, I suspect that juvenile outmigration rates remain low in Poudre Creek, as evidenced by the few individuals that were observed successfully passing the check dam, and that there might be opportunities to improve habitat for juvenile salmon. Reduced water velocities towards passage obstructions, whether they be a human-made dam, a road culvert, or a beaver dam, can cause obstructions in queues used by juvenile salmon to navigate downstream, which can cause migratory delay and ultimately increase the risk of mortality or predation. Freshwater outmigration of juvenile salmon is a particularly vulnerable period in their life history, but it is a primary driver in predicting the abundance of returning adults. Therefore, high mortality during this period, I think, will greatly limit the recovery of natal origin salmon in Lower Poudre Creek. In summary, through monitoring Chinook salmon in Lower Poudre Creek, we've identified some limiting factors or curiosities, if you will, that could depress recovery or resiliency in this nascent salmon population in Poudre Creek. Principally, these have been barriers to salmon egress and ingress. These selective pressures, while they definitely warrant further research, likely constrain spatial and temporal diversity, reducing the overall resiliency of the salmon run. But through identifying and naming them, we can begin to manage these threats adaptively. And in some ways, we already have. This year, folks at the Solano County Water Agency, namely the Streamkeeper, were able to, through collaboration with several organizations, remove the flashboards at the check dam earlier than they have in most other years and kind of find a solution that fit all parties. And so they were able to provide a greater migratory window for Chinook salmon in Poudre Creek this year. And with that, I'd just like to thank YBF's generous support of my research. Without programs like this, I wouldn't have had the opportunity to study these amazing fish, and I am so grateful. And if there's any time left, I can take any questions.
Thanks, Mackenzie. There is a question here. Has the Beaver Dam been breached to improve salmon passage?
Yes, to my knowledge, it has been breached. I think it was maybe last spring. I think last spring, folks from Solano County Water Agency took most of it down.
Have any other questions for Mackenzie and/or for any of our other guests? Okay, here we go, Mackenzie. They're coming in. Do we know how many salmon returned this year?
Yeah, so the surveys for this year haven't yet been completed. They're ongoing, but to date, I believe 28 salmon have been, or carcasses, excuse me, have been recovered. And there have been varying reports of how many live salmon have been counted. I think maybe the last report I heard was I think they'd counted 60 plus live salmon in the creek.
Nice. Is there any practical way manage flows to keep the dissolved oxygen levels in the creek above 7 milligrams per liter?
That's a really good question, and I think it's really hard. In Poudre Creek specifically, because we're positioned right below the Monticello Dam and Lake Berryessa, I think we have the ability to kind of manage our flows really strategically. And if we're monitoring dissolved oxygen concentrations in real time and we can see that they're dipping, and we have the flexibility to do so in that year, perhaps more water could be released to increase dissolved oxygen downstream. But we're much more limited in what we can do in the landscape more broadly, specifically in the tow drain, where a lot of that water comes from agricultural drainage.
And what time of year is downstream migration of the juveniles?
Juveniles migrate downstream in Poudre Creek anytime between March and late May, June.
What is the ideal timing for removal of the check dams? How does it change from year to year?
That's a great question. Obviously, I think that there's a lot of variability that we can't capture in timing of salmon runs, but generally speaking, fall run Chinook salmon are moving upstream as early as August. And there's data from a program run by the Department of Water Resources that indicates that there are adult salmon present in the tow drain, which is just downstream of Poudre Creek, as early as late August in some years. And so I know that folks are working to get, to work on passage remediation in lower Poudre Creek and open the creek earlier. But the up migration window for fall run Chinook salmon can start in August and can go to late December, January.
We get a nice comment for all three of you. Thank you all. So grateful for information and important research that you're performing. Let's see, back to the salmon here. Do you collect all the carcasses? Aren't they important food sources for other species?
Yeah, absolutely. They're important. But not only are they important food sources for other species, but they're a really important source of nutrients for the creek as a whole. And so we don't actually take any carcasses from the stream. We'll find a carcass, we'll collect the data that we need from it. So, things like length, the genetic sample, otoliths, coded wire tag if one is present, and then we'll put it back. So we don't actually remove any of the carcasses because they are so important.
Are there any areas of the creek where adults seem to die more often?
That's a good question, and I think it's really dependent on the year. Like in 2021, most of the adults died low in the system because of that low dissolved oxygen condition or that anoxic event. But generally speaking, in the past anyway, the salmon, when they can, are going to travel as far up Poudre Creek as they can. And so most of the time, that's why we find more carcasses kind of concentrated right below the Poudre Diversion Dam, because they'll go up as far as they can. And when they reach that dam and they can't go any further, that's when they'll make the decision like it's now or never. But generally you see more carcasses below pathogen impediments in Poudre Creek.
Are there many remnants of the Beaver Dam? And if yes, are they upstream or downstream of I-80?
I couldn't say. And beavers tend to be pretty industrious, so I'm not sure if they've started to rebuild. But when the Beaver Dam was taken down last year, I think they did a pretty thorough job of removing it.
And would removing bass increase salmon smolt survival significantly?
That is a good question. And I think the answer is we don't have enough data to say whether or not bass are contributing significantly to juveniles or to smolt mortality. Doing predation studies with acoustic telemetry is pretty difficult at this point with the technology where it's at. But there's a research project for you right there. I don't know if removing all the bass would improve survival significantly.
Well, again, thank you all. Thank you all for all of your research and information and sharing that information with us this evening. Like has been stated, very valuable for all of us to learn. I do want to remind everyone that where did I go? I lost us. There we are. That we have next month. There it is. The Flyway Nights on Thursday, January 5th at 7 o'clock. So if you're interested in learning more about the Fremont Weir and the Big Notch project going on up there, and how that's going to affect those of us who are downstream, that might be something that you would enjoy. Also, this evening's program is recorded, and I'll be working on getting that up onto our YouTube channel here in the next week or so. So thank you all. Thanks for joining us this evening, to our 3 students, and hopefully we'll see many of you again at future Flyway Nights.